Hello, my name is Ido Amit. I'm a professor in the System Immunology Department at the Weizmann Institute of Science, and I'm especially honored to be a CRI Technology Impact Award awardee. You know, technology is all about taking risks, developing something that hasn't been thought of or obviously used until today. And really one of the major problems in developing new types of therapies that would reactivate the immune system in cancer is to understand why the immune system is not active in the tumor site. And without actually understanding all of the parts that make the immune system and how they interact together, we would never have the capacity to understand how we can activate them, how we can use different types of therapies to activate the immune system against the tumor, to fight the tumor. And tumors really are very sophisticated in inactivating the immune compartment. And they evolved to be protected and shielded from the immune system from attacking it. And with the help of the CRI Technology Award, we've been starting to take very big risks in developing technology that would allow us to understand what are these exact mechanisms, how exactly is the tumor cells inactivating the immune system, making it exhausted, inactive. One of the, I think, most exciting technologies that we've developed is the technology called Zmansek. Zman in Hebrew means time, and so far when we've looked at the tumors, we looked at them in a way that we are capturing snapshots. So think of a movie where you only see one scene, and from that you have to deduce all of the relationship between the different characters. It's practically impossible. And our technology really makes that into a dynamic movie where we can see which actor met the other actor. So that type of capability, I believe, will allow us to, not in the very far future, develop much better ways to reactivate the immune system, especially areas that we have not touched until now. You know, we are trying to understand this complex movie and how all these players are coming together. And one of, again, the major players that were maybe missed or not captured to their full potential with these snapshots were macrophages. And macrophages, when they enter the tumor, they enter as, I would say, positive players who are immune cells that can attack the tumor. But very quickly, the tumor manipulates them into inhibitory macrophages, macrophages that are inhibiting the T cell response. And what we found is that there is a very special protein called TREM2, which is involved in this transformation. And we are developing different ways to block it in order to allow the monocytes or macrophages to become more active player in the activation of the immune response. And so far we're seeing really phenomenal results which are making us optimistic that this can be combined with the current T-cell checkpoints such as anti-PD-1 to make much more deep response for a larger number of patients. I think there's this battle with the tumor is a very challenging one because again the tumor has evolved to evade the immune system. I think we're learning more and more how we can reactivate the immune system again to fight the tumors. And I do believe that the more we understand the intricacy of the different ways that the tumor is blocking different subsets of immune cells, and the more that we can combine these treatments together, the more deep effects we're going to see, the more patients that are going to be responsive and even cured from tumor. The way we are seeing right now in melanoma, I'm really hopeful, and I don't see any reason why this same type of deep and effective response will not be in other tumors. There is no difference in the immune system of melanoma patient versus breast cancer or lung cancer patient. The only difference is we understand really well that movie, simple movie so far, whereas the breast and lung are a very complicated drama. And I think we're now starting to untangle this drama, understanding how the macrophages are playing their role and how another type of immune cells called dendritic cells, which are again the commanders, if you will, that activate the T cells, what are their missing role in such unresponsive tumors.